Hi everyone, this is Kayvan from University of Queensland. Today I'm going to present some of our research data on coconut rhinoceros beetles in the Pacific Island. So I would like to begin by acknowledging the traditional owner of the land on which we meet today, and also I would like to pay my respects to elder past and presents. So coconut rhinoceros beetles are serious pests of the coconut trees and other old palms in South Pacific Islands, and they are native to Southeast Asia, accidentally introduced to the Pacific Island almost 100 years ago, and they established their population in many countries. And in some countries, the damage of the coconut trees are seriously important for the people in the islands. So we don't have that pest in Australia, but it's a potential biosecurity risk for us because we have a great biodiversity of the palm trees in the North Queensland. So Oryctys rhinoceros nudibvirus is a large double-stranded circular DNA virus, something similar to baculoviruses, but it's slightly different. And they have been used as a biological control agent for almost 50 years. They have been found in the Malaysia and Philippines, and then introduced to Pacific Island in 1963, and then they kept the pest population under control for almost 50 years. But recent attempt to control the pest in new invaded country was successful, and they couldn't control the pest properly. And for the large scale of production of these viruses, they need some sort of cell line, but unfortunately we don't have any cell line from Oryctys rhinoceros. And there is another cell line, so from the proxy species, closely related species, which is black African beetles. And they use this cell line for the large scale production of this virus and release of the virus occasionally in Pacific Islands. So here we have a new wave of invasion started in 2007 in Guam, and then went to the Hawaii and PNG, Solomon Island, and most recently in New Caledonia and Vanuatu. It has been suggested that this range expansion is related to a particular haplotype of beetles, which claim to be resistant to virus, and that's why they escape the biological control and they invade a few new countries. But so far there is not enough scientific evidence to say that population of the beetles are resistant to virus. So we started our work with the screening of the genetic structure of the pest population across the region. We use genotyping by sequencing approach. We explore the nuclear DNA, and we also use mitogenome sequencing and the CO1 gene to just check the population structure across the region. Based on the CO1 gene haplotyping, we found three major haplotype groups in the region: CRBG, which known to be invasive and resistant to virus. We found that haplotype in Solomon Island. We found them in the native range of the pest in Philippines. We also found it in the New Caledonia. CRBS is another haplotype of these pests, which found in Tonga, Fiji, Solomon Island, and also in Samoa. And another haplotype group of these beetles is CRB-PNG, mainly collected from PNG, but we have this haplotype in Vanuatu and Samoa as well. So in some countries, we only have one haplotype group, but in some other countries, we have all three major haplotype group of these beetles. So the next step was checking the population structure based on the genotyping by sequencing. We sequenced more than 200 beetles from different haplotype group, and we explored more than 10,000 fragments of the genomic locations. And this GBS data resolved the population based on the different countries. For example, here we have Solomon Island in the top, we have PNG group in the bottom line, and we have Tonga, Fiji, and Samoa very close to each other in this corner. When we included the haplotype based on the CO1 gene, we can see obvious sign of admixture in the population here. So you can see population based on the CRB PNG and CRBG are located in the same cluster in the top. We check the structured plot and we can see some sort of gene flow and admixture in the population in Solomon Island and PNG. When we have admixture in the population, when these two different haplotype groups reach to sympatry in one place, from that point, we can't use anymore the CO1 gene as appropriate markers to study the invasiveness of these pests. So it has been claimed that CRBG is resistant to virus because previous group, they couldn't identify any virus in that particular haplotype. And they have done some sort of bioassay on the CRBG in Guam, and they couldn't get enough mortality by these viruses, which produce in the cell line, in the proxy cell line. So interestingly, we found high percentage of the virus positivity to the virus or infection rate, regardless of the haplotype group in all geographical population of these beetle. For example, here in Solomon Island, which are CRBG, we have almost, all of those examining beetle are positive to the virus. So we have a virus positive group and we try to see what exactly going on in this sample. We sequence using the nanopore technology, one individual of the positive CRBG positive to the virus. And we sequenced the full genome of the virus and we found some sort of point mutation. And we also found some deletion and inversion of the big fragment of genome of the virus when we compare this virus genome with the Malaysian isolate, which has been sequenced in 2008. So now we collected, we want to get a better picture of the population. We collected individual sample from Philippines, which is the native range of this pest, and the RCRBG. We collected from Solomon Island, which is invasive, and also the RCRBG, and collected the beetle from the established population of CRB PNG and also CRBS in Fiji. We used the Illumina RNA-seq technology and we sequenced more than 20 individuals and then infected and non-infected to the virus. We successfully assembled the virus sequence and we have a consensus of each population. And we found the gene expression profile of the virus and we compared this pattern in each individual in each country. And we also have some sort of single nucleotide variation, multiple nucleotide variation pattern or amino acid change for each population in 4 geographical regions. So interestingly here, we found the virus we have in Solomon Island is very similar to the virus we have in Philippines. So we believe there is some sort of co-divergence in the host and pathogen based on this phylogenetic relationship of the virus and also phylogenetic relationship of the pest based on the CO1 gene. So because the previous claims say there is no replication of the virus in the CRBG, we try to see if we can get immune response from the host to the virus infection. So we sequenced a small RNA sequencing and we found, interestingly, a large peak of the 21 nucleotide, which is a signature of the RNA response to the large double-strand DNA virus. We also found 1 microRNA in the viral genome, and we are working on this one at the moment to see how this viral microRNA can contribute to the host pathogen interaction. So our next approach was, sequencing of the transcriptome of the beetles infected and non-infected in different countries. We have the profile of these gene expression of the midgut in 4 different countries, and we found more than 1,000 genes differentially expressed in response to virus infection. And CRBG, we have Solomon Island and Philippines, but overall the response to the virus infection is different. For example, here we have more genes upregulated, but in the Philippines we have more genes downregulated. Interestingly, we found profound impact of some overexpression of the genes in response to the virus infection. For example, all of these antimicrobial peptides overexpress after infection with the virus, but the overexpression, the level of the expression in Solomon Island population, which is a newly invaded country, is significantly higher than other countries, which is very interesting data. And similar data previously reported from Lady Bird. So we have a sequence of the genome of these virus, these beetles. We have more than 20 transcriptome data, and now we know CO1 gene is not the appropriate marker to find the invasive population. So it's our job to find to suggest a new biomarker to identify the invasive population. And interestingly, we found some genes like, cyclopin, serpin, clotropsin, and few other antimicrobial peptides. They have a very low expression value in non-infected beetle in the new introduced country in Solomon Island when we compare them with the established and the native range population. So we are looking for some novel biological control agent, and we establish a pipeline for high throughput sequencing of the, analyzing high throughput sequencing of the data for virus discovery. We identified a novel picorna-like virus in the coconut beetles from Taiwan, but we couldn't find any trace of these viruses in other populations. So our collaborators in Taiwan are working on the impact of this virus on introducing of this virus to the population, see if they can get any immune response or how pathogenicity of these virus is going. So we have more than 1,000 RNA-seq data from 27 Scrabida beetles in the SRA database, and we would like to search these database for the virus discovery to see if there is any other virus in the similar insect. And we are looking for some collaboration with the other people to, if they are virus hunters, to get some sort of idea of the virus diversity in other Scrabida beetles. Okay, just as a summary, when we have a sympatry of the different haplotype group of the beetle in one place, the CO1 gene is not appropriate marker for, resistance to the virus or invasiveness of the beetles. And our data shows the CRBG haplotype is not solely responsible for the new wave of invasion, because we have CRB PNG in the Vanuatu, which is another newly invaded country. And overall data shows the importance of the immunity in insect pest incursion. And also I have to acknowledge our collaborator efforts in different countries. And this research is funded by ACR and also the University of the Queensland. I'm happy to take any question.
Well, thanks very much, Kayvan, for your presentation. And Kayvan, am I coming through clearly? I've had some connection issues. Can you hear me?
Yes, yes.
Oh, splendid. Looks, that's a relief. So Kayvan, a great presentation. I did catch the second two halves of that, and my apologies to everyone for being late, which is the Cardinal Sin as chair. We are pretty much bang on time. So what we might just do is, if there's a brief question, we'll throw one to Kayvan, but any longer questions or discussion might have to wait for the panel discussion a bit later on in our proceedings. So is there a quick question for Kayvan? Okay. It seems not, Kayvan, but maybe you'll get some questions later on in the afternoon.